In 2003, SARS spread across the whole world. In 2008, Ebola ranged in Africa. And during the next year, COVID-19 has caused many deaths and many activity has been called off. It seems that the tragedy never ends. Should we live in danger? It is time to stop. Stop! Although we cannot stop the virus from appearing, we can stop the epidemic from spreading. So, what should we do?
Rapid testing. Rapid testing can detect a virus in a short time. Thus, we can take prevention measures before the disease spreads. Currently, rapid testing commonly uses antibody to detect viruses such as HIV, norovirus, MERS, and so on.
However, there are some drawbacks of using antibodies. First, the cost of antibody is too high. Second, the antibodies are hard to purify. Third, the procedure of making antibody is time-consuming. Maybe we need something less expensive, faster, or easier to purify. Maybe a peptide work.
Working from the idea of using a peptide in a detection kit, we wanted to focus on dengue fever first. A total of 3.9 billion people in 129 countries are at risk of dengue fever. Almost half of the world population suffers from the threat of this terrible disease. Taiwan is also afflicted by dengue fever. In 2014 and 2015, this disease spread in Taiwan, and local community infection broke out, resulting in public panic.
Unfortunately, there are no detection kits produced in Taiwan. Instead of relying on foreign imports, we wanted to produce the detection kit by ourselves to increase the domestic testing capability. The product is our kit that detects.
We use Dendex to fight against dengue fever based on three-part strategy. First, we use a unique tech called linear array epitope to produce our peptide. Compared to the current method of the synthesizing peptide, LA can produce peptide on a large scale and the cost of the LA are lower. Meanwhile, we use Rosetta to model the interaction between the dengue fever and our peptide. Then, we use our peptide to produce the detection kit. We also did outreach to promote our idea to the people, raising the public awareness of the dengue fever.
Now, we would like to introduce the interaction between the dengue virus and human cell. Before producing the peptide, we need to understand the target protein. The dengue virus envelope protein, E protein in short, which is important in initial interaction between the virus particle and the host cell.
Also, based on previous research, we knew the glycosylation sites on the E protein. These glycosylation sites are the main sites of binding to receptors on the host cell. When the dengue virus enters a blood vessel, the glycosylation sites of E protein bind to a kind of receptor on the macrophage called C-type actin domain family 5 member A, CD5A in short. This interaction can allow the virus into the cell, leading to dengue fever.
In order to confirm that CD5A can bind to the E protein, we express the extracellular domain of CD5A and the E protein of the dengue virus, serotype 2.
Serotype 2 is the most common type of the dengue virus in confirmed cases.
Our biobrick is composed of T7 promoter, lag operator, thrombin site, E protein gene or CD5A gene, HA tag, link tag, and his tag. T7 is the most common promoter for protein expression in E. coli. We also use the lag operator to control the speed of T7 promoter. We add IPTG to the E. coli to induce the T7 promoter.
We also use thrombin to cut the unnecessary protein to ensure our protein has the correct structure. So our biobrick contains a thrombin site. The HA tag and MATE tag help purify our protein because we can use an antibody to attract it. The his tag can help us confirm whether the protein is expressed correctly.
The National Health Research Institute generously provides us a plasmid containing the genes for several proteins from the dengue virus. We amplify the E protein gene with the primer we design. This is the result of E protein genes. The picture shows that the size of E protein gene is 1,500 base pairs. After PCR, we ligate the E protein gene into the plasmid vector PT29A and PT29B. This picture indicates that the size of PT29A and PT29B are about 5,000 base pairs. The ligation and transformation of PT29A E protein were successful. After we confirm that the sequence size of plasmid is correct, we transform the PT29A E protein into the BL21DE3 and induce the protein with 1 millimolar IPTG for 2 hours. As you can see, the size of E protein is 70 kilodalton.
Simultaneously, since we need to prepare the CLAC5A sequence, we got the plasmid from the origin technologies and amplify CLAC5A extracellular domain and make take from the plasmid by using PCR. This picture shows the PCR result and the sequence size containing CLAC5A extracellular domain and make take is about 600 base pairs.
The ligation and transformation of PT29B CS5A were successful. This is the cloning PCR of PT29B CS5A. The sequence size of T7 promoter, CS5 extracellular domain, link tag, his tag, and T7 terminator are 904 base pairs, which is similar to what this picture shows. This suggests that our experiment was successful.
After we confirm the sequence size of the plasmid is correct, we transform PT29B CLAC5A into E. coli strain BL21D3 and induce the protein with 1 molarity IPTG for 5 hours. This picture indicates that our protein was successfully induced. As you can see, there are differences at the 33 kilodalton site between non-induction and induction samples.
After our protein expression was successful, we needed to ensure CS5A was expressed correctly, so we used a method called the Western blot with the his tag antibody to check it. As shown in the picture, the amount of the protein after the induction increased and the his tag antibody binds to it, suggesting that the induction was successful.
To produce peptides based on CLAC5A, we decided to use LAE. 3 of the most important parts of LAE were the design of oligo primer, template repeat PCR, TR-PCR in short, and adapt PCR, AT-PCR in short. Let's see the detailed procedure. First, we designed the TR-PCR and AT-PCR oligo primers we wanted. In primer design, we made the TR-PCR primers partially complementary to each other. On the other hand, we designed AT-PCR primers which contain the restriction site we need for later steps. Second, we took the TR-PCR primer to do TR-PCR. In the LAE protocol, TR primers are both templates and primers. Because they were complementary, we got many tandem repeat sequences, TRS in short, by extending the templates. Third, we took these tandem repeat sequences to do AT-PCR. At this step, we added the restriction enzyme sites on both sides of the sequences.
Our TRS biobrick was composed of T7 promoter, LAC operator, thrombin site, TRSS, and his tag. Next, we ligate the biobrick to the PT29B plasmid and transfer the plasmid into the BL21 to produce the peptide of TRS, PTRS in short. In our biobrick, we use the thrombin to cut off the unnecessary sequence at the peptide level, and we purify the PTRS using the his tag.
The advantage of the LAE technique compared to the chemical synthesis of a peptide is that LAE can produce peptide more efficiently. LAE can make a repeated sequence of the peptide chain. After cleaving the long peptide with the enzyme, we can get a lot of peptides. The chemical method cannot achieve this. compared to the antibody, a peptide is shorter and has a simpler structure. Thus, the production costs of the peptide are much lower, and the peptide can be stored for a longer time.
Now, let's see the result of TR-PCR. In this picture, we found that the lower the concentration of the primer was, the longer the sequences of our TR-PCR product could be. The higher latest mean much longer sequences to do TR-PCR. After several tests, we choose the best TR-PCR product to do AT-PCR, and the results are shown here. The bright letters are above 200 base pairs. We extract the gel and took it to do sequencing to check the nucleic acid sequence of our AT-PCR product. After we got the correct TRS, we transformed the PET29B containing TRS into DH5 alpha to amplify TRS. We confirmed our TRS size is about 210 base pairs with colony PCR and 13 primers added. The result of colony PCR showed that the size of TRS 151 is estimated to be 420 base pairs. We are still committing to the latter steps, hoping that we will get the expected results.
When making the peptides, we also need to do computer modeling and analyze the protein structures to ensure our receptor and ligand can interact. All the structures of peptides and protein, as well as their interaction, were modeled by Rosetta. Two methods were used to prepare the structures of the receptor and ligand. First, because there were only a few different sequences between our target protein and the crystal structure which was required as a template, we utilized Rosetta CM to generate the structure of the E protein from a local strand PL046 based on the crystal structure PDB1 or AN. Second, because there was no template and we designed the sequence by ourselves, we used the ab initio method to predict the peptide structures purely based on their sequence. Then, we utilized the clustering method to cluster the results and found the most probable structure of the peptide. Finally, we verified the interaction between these protein peptides and E protein based on the protein and protein global docking.
Now, I would like to tell you how we connect our project with the community. For a project to succeed in society, getting feedback from the expert is crucial. So we consult with some experts to improve our project. For instance, we got a better understanding of the process and the standard of the verification for medical device after consulting with the Taiwan Food and Drug Administration. This helped us figure out what we should do in order to apply the permission to produce and sell our teeth in Taiwan. The Dengue Prevention and Control Center gave us the advice about pretreatment of the blood sample. They suggest that we use whole blood to more convenient detection on site, especially in the undeveloped and remote area like a centrifuge. Kaohsiung Medical Universe Zhonghe Memorial Hospital expressed the willingness to collaborate with us, providing serum for free tests if our product was complete. Moreover, we made great effort to educate the public about science and raise awareness about dengue fever. We want to maximize our impact to reach the people for various groups, age, and background. Thus, we engage the diverse group of people using unconventional network. For instance, we went to Mingyang High School, which is a juvenile correction school. These students are quite alienated from education, especially science education. Thus, we hope to disseminate scientific knowledge to reduce their awareness of science. Moreover, we create the children's storybook to spark children's interest in science. In order to reach general public, we create a podcast program talking about synthetic biology. Furthermore, as Taiwan is the aging society, This is essential to reach the elderly community to disseminate science. We try to break down accessibility barrier to science by disseminating information about our project in Taiwanese Hokkien, the language spoken by the majority of the elderly. We also recognize the opportunity in integrating arts with science to broaden social awareness. Thus, we work with the performing arts troupe to use glove puppetry, which is part of charming traditional culture of Taiwan. The convey science knowledge is to large audience.
Using the strategy described above, we produce our own detection kit. Our detection kit is similar to a commercial pregnancy test, which has a control line to ensure the product is reliable and a test line to indicate a positive result. We produce two peptides, which could bind to the E protein. The first peptide, PTRS1, was attached to gold nanoparticles and placed on the conjugated pad. The second peptide, PTRS2, was deployed on the test line. E proteins from the dengue virus were attached to the control line. If there are no virus particles in the sample liquid, PTRS1 conjugated on the nanoparticles will not be attracted to PTRS2 in the test line. Thus, they proceed to the control line, where a red band appears due to an aggregation of gold nanoparticles attached to PTRS1 as they band with the E protein there. If there are virus particles in the sample liquid, some PTRS1 will bind to the virus particles on the conjugated pad. When reaching the test line, the unbound site on the virus can interact with PTRS2 attached to the test line. A red band appearing on the test line indicates a positive result. Residue PTRS1 then flows to the control line, so a red band will also be found on the control line.
In the future, we hope the PTRS can be applied in the medical treatment. Because the PTRS can bind to the E protein of the dengue virus, it could be injected into the body to block the interaction between the CD8 on the macrophage and the E protein on the virus particle in the blood. Further hope our kit will be expanded to detect all 4 serotypes of dengue virus. Furthermore, it will also be able to do the environment detection to detect the dengue virus in the mosquito vector. This strategy of fighting dengue fever could also be used to fight against other diseases. As long as we know the structure of the virus and the partial sequence of receptor, we can use modeling to predict the probable binding site. Then we can use the LAE to produce the peptide of the predicting binding sequence. Finally, we will promote our idea and product to the public. By using this strategy, we may be able to fight against the disease in a brand new way.